Good morning. So my name is Roula Abdel Massih. I'm a little shorter than the previous speaker. Okay, so I'll be talking about the antibacterial and anti-proliferative activity of different forms of citrus pectin. As you all know, for centuries, plants have been used as remedies and treatment for diseases. And as we've seen in most of the talks, plant-derived chemicals represent a vast untapped source of medicines such as antimicrobials, antioxidants, anti-cancer, and we see that there's a lot of more exploration needed in that sense. In our lab, we worked on the antibacterial activity of different plant extracts, such as Rhumra ponticum, Olea liripia, Viola odorata, Eryobotria japonica, and others on highly resistant gram-negative bacteria that we've obtained from clinical hospitals. We also tested some of these extracts that had activity against gram-positive bacteria such as Staphylococcus aureus. And we found some apoptotic and anti-proliferative activity of certain extracts of Origanum majorana on human leukemic cell lines. We found antioxidant and anti-proliferative activity also in Olea liripia, olive trees. And kind of more relevant to my talk today, we found some antibacterial activity in pectin. I'll be talking about pectin today. What is pectin? As some of you know, pectin is a branched heterogeneous polysaccharide found in the cell wall of plants and in a variety of fruits. We say that it's heterogeneous because pectin is really a general term. It's not one structure. It's made of different components joined together. For instance, you have homogalacturonan, which is the main component. It's made of galacturonic acid backbone with certain methyl and acetyl groups attached. We can have modifications of that where you have xylose side chain giving you xylogalacturonan, or certain kind of more complex side chains giving you rhamnogalacturonan 1, or you can have attached to them a completely different backbone which is made of alternating galacturonic and rhamnose side chains with neutral side chains such as arabinan and galactan. The structure of pectin varies even within the same plant. It also varies between plants and the species, the age, and the maturity of the plant parts. As a function in a plant, it's involved in assembly, cell wall structure, adhesion, porosity, fruit development, and others. And it has certain applications, such as it's involved in eliminating toxic heavy metals, as shown in certain literature papers. It's a reservoir for biologically active oligosaccharides. It's used in industry as a stabilizer or thickening agent. I think you know pectin solidifies and kind of forms gels, or it's involved in jams. It's involved in reducing serum cholesterol level, decreasing heart disease, reducing intestinal infection, promoting the growth of healthy microflora. We heard yesterday a lot about prebiotics and probiotics and how basically at the end of the day pectin is a soluble fiber, so it can help in that sense in allowing the bacteria to grow. And certain papers have found certain effects of cancer, certain effects of pectin against cancer, depending on the cell line. The low solubility of this pectin and the complexity of its structure kind of limits its use in medical field. The main problem is the difference in structure. Every time you extract it, depending on how you extract it, you can end up with completely different products. So scientists have developed what we call modified citrus pectin, which is kind of a lower molecular weight. It can be produced either through pH modifications, so you have alkali and then acid treatments, where you break part of the pectin and you get a lower molecular weight. Basically, you're getting also rid of certain side chains such as arabinans, and you're exposing the galactans and arabinogalactans that are thought to have a role against cancer. You can also do similar things through heat treatment or enzymatic reactions. The modified citrus pectin I'll talk about today was produced through enzymatic reactions, so kind of a more structural, known structure. The MCP and cancer in literature has shown to have certain biological activities, such as blocking angiogenesis. We also heard about angiogenesis yesterday, and how blood capillaries can grow towards a tumor, so it can block that. It retards metastasis by blocking certain lectins, specifically galactan-3. So since it has galactans, and you know certain cancer cells might want to bind to the galactose, so they're blocked by this pectin. It promotes apoptosis or programmed cell death, and in some cases it was found to act synergetically with chemotherapy. So what's the aim of this work? It was to study the anti-proliferative and antibacterial activity of citrus pectin that we've extracted and the modified citrus pectin, specifically that was enzymatically treated, pectasol. So in the first part, to study the anti-proliferative activity, we chose to study an in vitro skin cancer model. We studied the Hackett cell line, and basically we studied the cytotoxic effect of citrus pectin MCP on the cell line, the anti-proliferative effect, and we tried to relate the anti-cancer activity with antioxidant activity. As some of you know, this is how you go. You can go from the keratinocytes through mutations to the Hackett cell line, and then through certain mutations, you can form the benign tumorigenic A5 cell line, and then the malignant RAS24 cell line. I'm going to show the results on Hackett. We're still doing the experiments for the rest of the cell lines. But what is interesting about this model is that you can look at kind of the progression of the cancer till you have metastasis. So just skip this stuff. So basically, to be able to study that, we studied cell viability using the trypan blue exclusion test. And here you add the trypan blue dye. For those of you familiar with this, you know that when you add this dye, cells that have an intact membrane don't take the dye, whereas cells that are dead will take the membrane, and you basically do a cell count. You can have a more kind of quantitative result through cytotoxic activity by studying the lactate dehydrogenase or using the lactate dehydrogenase assay. You can study the anti-proliferative activity of these cells using this chemical through the WST1 proliferation reagent. We had a speaker yesterday using the MTT, which is similar. So basically, you're monitoring the mitochondrial activity and seeing if cells still have ability to proliferate. And we also studied the antioxidant activity using the DPPH assay. So some of our results, this is the effect of citrus pectin on the viability of the cell line using the trypan blue assay. We see this is the negative control. You have 100% viability. Positive control, Triton X100 killed the cell. There's no viability. And this is pectin at different concentrations. You can see that there is somehow a dose-dependent effect. It kind of plateaus at the end. But at 750 microgram per mil, we could not, at the conditions used, add more pectin because it would solidify and form a gel. We did not have a more decrease in viability. So we kind of used the LDH assay. We again see a dose-dependent effect, and basically cytotoxicity reached around 40%. We tried this with the modified citrus pectin, the smaller with more exposed side chains, and we see a better result here. With trypan blue, we see a dose-dependent result, and we can kind of determine IC50, or the concentration at which 50% of the cells are killed. So it's between 300 and 500 microgram per mil. Using the LDH assay, it gave us a more kind of precise measurement. We can see the dose-dependent result again. So the MCP could kill the Hackett cells at 50% of them were killed at 500 microgram per mil, and we can see that at 750 microgram per mil, we had around 85% cytotoxicity, which is promising. We studied next the anti-proliferative activity of pectin and MCP. For pectin, again, there was a kind of decrease in proliferation, but it was not more than around 30%. Whereas with MCP, we could see that at 300 microgram per mil, which is here, we have around 50% decrease of proliferation. At 500 microgram per mil, we even reached 70% anti-proliferation. So this kind of gives us that MCP has more anti-cancer activity. Of course, more work is needed on how they're being killed, but it has more anti-cancer activity than the citrus pectin that we were testing. We wanted to see if that also relates to more antioxidant activity, and we used the DPPH assay, where basically a decrease in the absorbance indicated that your chemical is basically scavenging or reducing the DPPH radical. So we used different concentrations of pectin. Using this assay, we could reach around 4 milligram per mil. We see that there is a kind of dose-dependent response, but scavenging activity is kind of not very high compared to the positive control. If you convert that to antioxidant activity, we see that we reached only around 16% antioxidant activity. If you plot these two together to see if we have IC50, which is the point of intersection of the scavenging and antioxidant, we see that we could not reach it with pectin at the concentrations tested. So we repeated that with MCP. We can see that we have a dose-dependent scavenging effect. Antioxidant activity kind of reached 40%, which is better than that of pectin. And IC50, which is the intersection of these two graphs, could be reached between 2 and 4 milligram per mil. So it's not very high antioxidant activity, but at least we know that it's more than that of pectin. So as kind of a conclusion for the first part, MCP was found to be more cytotoxic than the citrus pectin. We had an IC50 of around 300 microgram per mil, whereas we could not find that in pectin. Also, the anti-proliferative activity of MCP was greater, and the antioxidant activity was also found to be higher than that of pectin, where we found IC50 between 2 and 4 milligram per mil. And the second part we wanted to study, before that, and our conclusion was basically that this different effect is not only due to the molecular weight of the pectin and MCP, but also, of course, due to exposing certain side chains, such as the galactan side chains, which have been shown, and there are certain studies to try to look for chemicals with these galactan side chains so that they can bind to galactans and stop proliferation. The second effect was to test antibacterial activity of citrus pectin and modified citrus pectin. We studied the minimal inhibitory concentration and the minimal bactericidal concentration of citrus pectin against different strains, clinical strains of Staphylococcus aureus. We also used a ATCC strain. And we see that the minimum inhibitory concentrations, they basically ranged between 0.39 and 3.125 milligram per mil, which is good. And the MBC ranged between 3.125 to 12.5 milligram per mil. That of MCP also, it had good MICs and MBCs. They're comparable. It's a little bit higher MIC, 0.78 and 12.5, but the MBC were comparable. They're the same. So to try to study that further, we tried to do a checkerboard between MCP and the antibiotic that was usually being administered for these clinical strain, which is cephalothin. And the way you do a checkerboard is you take the 96-well plate and you do serial dilutions of each of the antibiotics tested. So you take cephalothin, and here you have no cephalothin present, whereas in the rest of the wells you place a serial dilution of those. Then on the same 96-well plate, you do the same thing, but with your second chemical being tested here, it's modified citrus pectin. And basically, here you have only MCP, and all the rest, they are mixture of antibiotic cephalothin and MCP. And the mixtures that are kind of highlighted in red, basically they indicate the minimum inhibitory concentration. So minimum inhibitory concentration according to the cephalothin antibiotic is 0.5. The minimum inhibitory concentration for MCP by itself is 3.125. And what is interesting is that you see when you mix them together, these yellow kind of wells show you that there is a decrease in MIC, okay, which would indicate that if you have a very toxic antibiotic, you can supplement or use certain supplements to basically decrease its toxicity and still have the same effect. So what you do next is you calculate the fractional inhibitory concentration for each one. So you take each concentration that had no growth and you divide that by the MIC. You get what we call fractional inhibitory concentration. You add these fractional inhibitory concentrations, you divide them by the number of FICs that we have. You get what we call mean of sum of FIC. I did that for 25 strain, and for the strain that I'm showing here, it's 1.53. You need to plot these together, and basically what you have is that if the mean of sum of FIC is between 0 and 0.5, then these two chemicals tested are synergetic. If it's between 0.5 and 2, then we have an additive effect. If it's between 2 and 4, then it's kind of indifferent or neutral. More than 4, it's antagonistic. So what we got here is that we have only few strains on the border, if you want, of synergy, but most of our strains show what we call an additive effect of MCP and cephalothin. We're still trying to do tests with different antibiotics using pectin and this drug. So as a conclusion, the antibacterial activity of pectin, as a conclusion for this part, basically varied according to the source and molecular weight of pectin, according to the pH. I don't show the pH results here, but we studied pectin and MCP at different pHs, and we saw that their activity was optimal at pH 6. They both exerted a strong antibacterial and activity, both MCP and pectin, and the checkerboard results show that they are basically mostly additive. Okay, so with this, I would like to thank you, and if you have any questions, this is basically my university in Lebanon, Belamant. Thank you.
Any questions out there? All right, got one in the back.
I found this lecture really interesting for me. I got a question. I mean, I have a lot of questions, so actually I can ask after, but first of all, I would like to ask you why you choose the DPPH test for antioxidant capacity.
Sorry, why did we choose?
The DPPH test.
Yes, we were using, I wasn't the one doing the DPPH assays, but we had different antioxidant assays. We found that they were giving the similar results, so that's why I showed the DPPH assay, okay. But they've done different tests for antioxidant activity.
So the results were quite similar, or?
Yes, the results were quite similar. We have average antioxidant activity, though literature shows that there's more, that pectin has a very strong antioxidant activity.
Yeah,
and the pectin that we got, there was kind of average antioxidant activity.
Okay, and can you provide me the details? How about the extraction procedure? I mean, you extract.
Yes, I cannot tell you how the citrus pectin was extracted because we did that in collaboration with Megazyme, which is a company. The MCP we obtained from the market Pectasol C. So you can get that if you're interested.
So it's commercial one? Or?
The Pectasol C, yes. Okay. The other one, we cannot say how it was obtained.
Okay, thank you very much. Okay. And the rest of the question
yes,
I will ask you.
Thank you.
Thank you. Good talk. I have two questions. One is, DPPH assay is good for general antioxidant measurements. Have you tested any specific hydroxyl radical or SOD type of activities?
Yeah, that's what I was answering. I didn't do the antioxidant activities, I did the rest, but yes, my colleague did several antioxidant. I chose this one because I've worked with it before, but they have all similar results.
Okay. Yeah, the next question is, if you are to apply this in, I know it's a long way, if you are to apply in this clinical setting, what would be the human equivalent dose for antioxidant and antibacterial activities? What you guess?
Pectin has already, pectin as general, but as I said, pectin is really a very common name. It's like saying a certain sugar. There's so many different types of pectin. Yeah. But pectin, before even doing any trials, a lot of people just are using pectin all over the world, especially against prostate cancer. Because it seems that it is approved to be used because it's found normally in citrus and in apple fruits. They started using that, I think, 10 years now, and they're finding positive results. But now all scientists are actually doing the lab work to kind of prove that it has certain activity. We're more interested in the antibacterial activity because not much work has been done there, and we're finding very promising results. Yani our results are showing us sometimes things that are as high or almost as high on very resistant strains. Yani we're using strains that your antibiotics sometimes are not working on, and I didn't show these here because I was just trying to show a general view. So pectin and cancer, there's a lot of work, but as I said, we still are interested in that because pectin is a general or generic name. There's a lot of pectin in the market. You have pectasol, you have GC100, and each one represents a different molecular weight, different structure. Some are more methylated, more acetylated, and each claims different roles. So we wanted to do a kind of systematic approach to kind of find the basic science for that.
Thank you.
That's it.
Any more questions? If no, let's formally join me in recognizing one of our nice talk.
Thank you.